Hi, my name is Rebecca Ahrens-Nicklas, and I'm a physician scientist at the Children's Hospital of Philadelphia and the University of Pennsylvania. And today I'm going to be talking about how CLN3 disease disrupts the function of neuronal networks in the brain. So, when we think about how we study lysosomal storage diseases like CLN3 disease, we have to start thinking about what we are missing in order to understand how we can move forward the field and hopefully develop meaningful therapies. There's a lot known about CLN3 disease based on decades of work, including a lot known about the natural history, so what actually happens to the patient. We know that it's a disease first characterized by blindness and then progressive dementia, neurologic dysfunction, and regression. We know what the gene is for CLN3 disease. It was identified in the 1990s. There's been a lot of really robust and important studies trying to understand what happens at the cellular level when you lose the function of CLN3 protein, including some recent groundbreaking work showing that you can have accumulation of lysosomal glycerol phosphodiester species in the lysosome when you lose the function of this protein. But one level that historically has been understudied is what happens at a network level in the brain, right? The symptoms that patients suffer from, whether it be cognitive dysfunction or seizures, result from dysfunctional firing or dysfunctional behavior around networks of neurons that essentially drive disease pathogenesis or drive the disease symptoms. And so one aspect of CLN3 disease that my group has been working on over the years is to try to understand at a network level what happens in the brain of CLN3 disease models. So today in my talk, I'm going to provide an overview of a number of emerging lines of evidence suggesting that neuronal network level function is disrupted in CLN3 disease. Starting with our work just characterizing this dysfunction in mouse models, and then talking through what cell types we think drive these circuit level changes. And then finally, focusing on if the network dysfunction can be corrected, and importantly, how early do these changes arise? So we can try to guess or understand how early we might have to intervene if we're going to prevent this network level dysfunction. So historic or previous work has been done looking at synaptic transmission to try to get a sense of neuronal excitability in mouse models of CLN3 disease. And this is beautiful work that had been done by Benedict Grunwald, shown here. And his group had shown that in middle to late stage disease mice, so about 14 months old mice, you see differences in both excitatory and inhibitory neurotransmission. And this is data shown here in the hippocampus, but they also saw differences in the amygdala and the cerebellum. And it looks like there might be specifically more difficulties or more vulnerabilities on the presynaptic side of the synaptic neurotransmission. But basically, these data suggest that whatever the biochemical difference is, whatever the loss of CLN3 protein is doing, it is leading to differences in how the synapses work and interact and fire. This suggests that if you then scale this across many synapses, you're going to have differences in network level excitability in the brain. So building on this work, we sought out to try to understand and characterize at a whole hippocampus level what type of excitability differences we might see in mouse models. And the reason we picked the hippocampus to start with, because it's a network in the brain that's important for learning and memory. And we know this is a network where CLN3 protein is expressed in the postnatal brain, at least in animal models. Practically speaking, it's also a network that you can study using well-validated tools. And one of the approaches or methods that we use in our lab is in vitro voltage-sensitive dye imaging. So essentially what this is that you actually take slices of brains from CLN3 mice, and then you put them in a dish and keep them alive and load them with a dye that fluoresces with voltage change. You then stimulate the slice, so you essentially provide an electric current to the slice in order to get the circuit to fire, as sort of modeling what would happen if input came into the hippocampal circuit in vivo. And then you record the voltage at a very high frame rate because these electrical differences change very quickly over time. And then we analyze the data. And this figure here is just showing you a model or a diagram of the hippocampus. And you can see here, when you stimulate at time minus 10 milliseconds in panel C, after you stimulate at the time zero, you see this robust excitation of that first part of the hippocampus, the dentate gyrus, shown here in red. And we can do these and try to understand how the disease changes the excitability of the network. When we did this many years ago at this point, in early stage mice, what we found is that the hippocampus becomes hypoexcitable very early in the course of CLN3 disease. And this in the first column is just showing you in wild type mice at 2 months of age, and then down below at 6 and 7 months of age. The asterisk is just the part of the circuit that we're stimulating. And what you can see is we stimulate the circuit 4 times, and you see 4 different activation fronts that spread across the entire dentate gyrus. But when you look in a diseased mouse at 2 months or 6 months of age, it just isn't as robust excitation. So it's a little bit more yellow, a little less red. If you then do statistical analysis to be able to compare the differences across the entire circuit over time, you can see that things shown in blue here are statistically hypoexcitable, suggesting that very on in the disease course at 2 months of age, that the hippocampus is hypoexcitable. And this is long before historically we have thought that there's significant amount of, for example, of storage accumulation or neuronal death in these mouse models, suggesting that function is affected very early in the disease course. Another way of measuring function in vivo is through EEG analysis. So we implant electrodes into the mouse brain. The mice are allowed to recover and then recorded for 72 hours. The EEG gives us an idea. Essentially, we process this EEG signal to give us an idea of both how background activity levels of the brain might be changing over the course of the disease, but also if there's any sort of epileptiform discharges, things called spikes, that would tell us whether or not these mice have a propensity to seizures. And we do a variety of different analyses, including artifact detection to make sure we're looking at good signal. And then we have automatic spike detectors. And then we do power spectrum analysis using something called fast Fourier transform analysis. All this means is that you take an EEG signal and you figure out how much of that signal is composed of slow components versus fast components in different frequency bands. And this gives you an idea about the background resting state of the brain. And when you do this in CLN3 mice, and these are aged CLN3 mice that are later in the disease process, we see some differences. We see both differences in that background activity, so how much slow versus fast activity is present on the EEG, and differences in those spikes. So if you take a look here in this first panel, you can just see some raw EEG data from the wild type mouse versus a CLN3 mouse. And where those asterisks are representative of epileptiform spikes. And if you quantify how many spikes are happening over a 30-minute period, we see a lot more spiking activity in the CLN3 mice, suggesting there's abnormal network level activity that are leading to excessive discharges. And these spikes or these epileptiform discharges could provide a substrate for seizures. In the last panel, what you see here is a quantification that we do to see how much slow versus fast background activity there is. And we don't need to understand all the details of the different frequency bands, but what you can see here is that first set of bar graphs, those first bars on the graph, quantify how much slow activity there is. And as you can see, in the CLN3 knockout, there's less slow delta activity and more activity in some of those higher frequency bands, suggesting there's a shift towards faster background activity in the EEG. Again, suggesting that there's abnormal differences, or there are differences in how resting state connectivity, resting state excitability is progressing in the context of CLN3 disease mice. So overall, from a lot of work, we found that network dysfunction using both EEG and voltage-sensitive dye imaging starts very early in mouse models of CLN3 disease. It's progressive. We have validated in multiple models of CLN3 disease. It's similar in some of the features of the EEG backgrounds to what's seen in some more common forms of epilepsy. In addition, it also shares some features with Alzheimer's disease mice, which is intriguing given the commonality between Alzheimer's and CLN3 disease in that they're both, progressive dementia disorders, suggesting that whatever network level mechanisms that underlie CLN3 disease might actually mimic or sort of mirror what happens in adult onset neurodegenerative conditions. So the next question we wanted to understand is to understand what cells are driving this network level dysfunction, right? We see that the circuit is broken And the question is there one cell population that's driving this dysfunction, such that if down the road we wanted to try to develop a therapy, we would know what cell type we really had to target in order to be able to make a meaningful difference at a network level. And so to ask some of these questions, we utilized a new conditional rescue model mouse that was initially developed by Beverly Davidson's lab, our close collaborator and friend. And essentially what this mouse model allows us to do is that on the background of a CLN3 disease mouse, in this case, the Delta 7/8 mouse that was developed by Sue Kaufman, another allele was introduced that's a floxed full-length transcript of the CLN, or full-length copy of the CLN3 gene that's in the reversed orientation. When it's introduced to a Cree driver line, essentially you excise those LOXP sites and you can reverse the orientation of the CLN3 transgene and it is turned on. So essentially this allows you to rescue the Delta 7/8 mouse model using specific Cree drivers. So you can start to think about rescuing at different points in time, or in this case, different cell types. So in order to understand which cell populations need to be rescued in order to normalize network level dysfunction, we use two different Cree recombinase driver lines, a GFAP Cree model that rescues expression in astrocytes, and a SYN1 Cree model that rescues expression in neurons. So what this allows us to do is say, okay, if I just rescue the neurons, is that enough to fix the physiology? Or if I just rescue astrocytes, is that enough to fix the physiology? We then went on to do histology to look at things like storage accumulation and other hallmarks of disease, and then our markers are our readouts of network level dysfunction, including voltage-sensitive dye imaging and EEG. So what we found is that when you rescue in either astrocytes or neurons alone, you do not fully correct storage, right? So this is showing the hippocampus on the top and the motor cortex on the bottom. The first column shows you a wild-type mouse. The second column shows you a diseased mouse, and you can see in green the accumulation of subunit C of the mitochondrial ATP synthase, a hallmark of CLN3 disease storage accumulation. This is on the top looking at the dentate gyrus and hippocampus. And so when you look at the GFAP rescue, so astrocyte rescue mouse, or the SYN1 rescue, neuronally rescued mice, you still see a lot of green, telling us that rescuing in one cell type alone will not prevent storage accumulation. What about other aspects of histopathology? Another marker of disease pathology as the disease progresses is a reactive astrocytosis. We know there's inflammation in the brain of CLN3 mice and CLN3 patients, unfortunately. And what we found here is similarly, like we didn't correct storage accumulation with just rescuing CLN3 expression in either astrocytes or neurons alone, we do not fully correct astrocytosis. So we're not fixing the storage completely. We are not fixing the immune response to that storage completely. What about physiology? Interestingly, when we start looking through voltage-sensitive dye imaging, we see some really interesting patterns emerge. First off, this is just showing you what in a Delta 7/8 mouse model, a heterozygous Delta 7/8 mouse model, as compared to a fully diseased Delta 7/8 homozygous mouse, that we see significant hypoexcitability of the dentate gyrus. These data are very similar to what I showed you earlier in a different CLN3 mouse model. So this is if you have no CLN3 rescue, so no Cree driver. What happens if you use that GFAP Cree driver to rescue expression in astrocytes alone? What you can see here in that last graph, there's still areas of blue. So you're still having areas of hypoexcitability resulting from essentially or indicative of abnormal network dynamics in the brain. Now, if you rescue in neurons using that SYN1 Cree driver that I spoke about, we see full rescue of hippocampal excitability. If anything, it becomes a little bit more excitable, even though there was no correction of the storage or astrocytosis. This tells us that CLN3 is playing a role in neurons when it's doing something to change or ameliorate or prevent differences in excitability in the neuron. And those differences might be independent of the storage phenotypes we see or the reactive immune changes we see, suggesting that CLN3 itself might in some way be contributing to synaptic function or synaptic excitability in neurons. How about via EEG? So I showed you the data from voltage-sensitive dye imaging. What about with EEG? You can see here, we see the same background slowing. So essentially that loss of delta activity in the diseased mouse. When you rescue expression in astrocytes, we do not see correction. So essentially they don't look any different than the diseased mice. But when we rescue expression in neurons, we see restoration of normal EEG patterns, again suggesting that if you turn or rescue CLN3 expression in neurons, you are somehow modulating synaptic transmission, you're somehow modulating expression or excitability such that you normalize network level function in the brain. So it really tells us we should be going after neurons when we start thinking about correction of CLN3 disease. One last figure from our EEG data looking at epileptiform spikes. So those extra discharges, those seizure-like activities, you can see they're increased in our mouse model versus wild type. Interestingly, if you rescue expression in astrocytes or neurons, either of which is sufficient to support this extra activity, suggesting that CLN3 and astrocytes might be playing a modulating role in terms of sort of damping down any extra activity or extra epileptiform spikes that might be arising in the setting of disease. Okay, so that was using a genetic rescue approach to try to correct network level differences in CLN3 disease. But for practical purposes, if we're going to try to move forward trying to think about a therapy, we need to start thinking about other ways we can replace CLN3 expression. And the obvious method to do that with currently available tools is through AAV gene therapy. This is something that's been well studied in CLN3 disease. And we wanted to see if AAV gene replacement would be sufficient to normalize some of these network level differences that we've been identifying in our mouse models. And so this is just showing you our CLN3 AAV construct, which actually uses a segment of the endogenous promoter to drive somewhat physiologic levels of expression. And as you can see here, our red in panel A, the Delta 7/8 mouse has very little or no expression of CLN3. But with AAV treatment at P0, we're able to restore about 25% of heterozygote levels. And that level is sufficient to partially decrease the amount of subunit C accumulation shown in B and C. So we're seeing expression with our AAV gene replacement therapy, and that expression is having some impact on the histology. But what about function? So if you take a look at P0 delivered AAV gene therapy at this low level of expression, we were excited to see that you were able to fully rescue both the spiking frequency. So remember those extra discharges that might be a sign or symptom of epilepsy or a propensity to epilepsy, shown in gray and C. And then in D, we also normalize those background frequency bands, suggesting that early rescue of CLN3 expression in the at least in markers of hippocampal dysfunction and can indeed normalize function. So we then moved on to say, okay, in early stage disease, 2 months of age, for example, we saw differences in excitability of the circuit. And this is actually much earlier than most people had thought we would start to see disease phenotypes in a mouse model of CLN3 disease, which we all know these mice have very mild phenotypes. In the human condition, patients don't start having symptoms of the disease until, beyond preschool years, sort of in early childhood. And so we really didn't know if biochemical markers of disease or other features of the disease would be present essentially from birth. And we wanted to start to understand if these markers were present because it would have real implications for the timing of which you might try to apply a therapy. And so the first thing we just wanted to know is that in very early stage mice, in this case P11, whether or not we were starting to see those hallmark biomarkers of disease, glycerophosphodiesterase that were recently identified, accumulating in CLN3 mice. And what we saw is that throughout the brain, cortex, hippocampus, cerebellum, in CLN3 knockout mice, we saw a significant increase in the accumulation of glycerophosphonolactone. And this is in whole brain homogen or in brain homogenates. This is not through lyso-IP and looking at metabolites in the lysosome. So you can detect it in brain homogenates itself. Other glycerophosphodiesterase were not as abundant or not as accumulating at this early stage. It's possible that if you looked in the lysosomes specifically, you would see the accumulation in lysosomes at this early stage also. But what we think this tells us is that this change in GPI starts early and probably contributes in some way, at least, to the excitability or synaptic dysfunction that we're seeing. And so we really wanted to quantify the earliest stages of network level dysfunction in this mouse model, because if you start to think about the timing of, for example, AAV gene therapy, it is not clear to me, in all honesty, that if you give an AAV gene therapy to an already diseased circuit, that you can remodel that circuit, that you can gain back essentially those network level differences that you've identified. So it's really important that we figure out how early that circuit level dysfunction begins. So we went back to our model of using voltage-sensitive dye imaging, but this time taking slices from much younger mice, starting as early as postnatal day 7, shown here at the top row that's shown on the screen. And what you can see here is that these graphs are oriented just a little bit differently, but essentially where the white asterisk is where we're stimulating the dentate gyrus, and we're looking at spread of that signal through the dentate gyrus and then down through the rest of the hippocampus. And I hope you can appreciate that the CLN3 knockout mouse is very hypoexcitable. So essentially, you stimulate, you give the same amount of stimulus to both slices or both groups of slices, and you really just don't see that much response, not much red. And the areas shown in purple on that last graph are the areas of statistically significant differences between the two circuits. So this is what happens at P7. You move forward to P14, and it looks like there's actually some compensation, right? So it's sort of the circuit tries to reset itself as the hippocampus is maturing. So this period from P0 to P21 is a time period where this circuit changes rapidly during early postnatal life. It matures to its final adult state. And you can see at P14, it does an okay job in sort of catching up, right? So the differences are not that profound. But then by P21, that maturation and that ability to catch up and to normalize circuit function has failed. And essentially, we have now set up what we saw in our previous paper that by 2 months, you have this hypoexcitability of the circuit. So if you look at the bottom line, the P21 line, what you're seeing essentially is that the CLN3 knockout hippocampus is hypoexcitable as compared to controls, which is what we saw at 2 months and 6 months and on down the line. So this tells us that as early as P7, 7 days postnatally, which in reality, if you convert that to sort of a human timeline, that's about birth for a human, there already are differences in the excitability of the circuit, suggesting that whatever CLN3 protein is doing, it's affecting synaptic transmission very early on. This is now showing you the rest of the circuit by stimulating Schaefer collaterals. And again, we see differences very early on. There seems to be a compensation that just can't be sustained, suggesting that all of these changes start very early on in life. What about in vivo at this point, right? So I showed you the voltage-sensitive dye imaging data. Now we do EEG recordings, and I'm showing you EEG leads from the region of the hippocampus, shown where that electrode is in part A. And essentially what we see is that even as early as P30, we see an increase or a propensity towards increased spiking activity, and we see differences in the background frequencies. The differences are a little bit different than you see in aged mice. In this case, at the very early stages, you actually see EEG background slowing with an increased delta power before you reach that phase later on where you lose your delta power, suggesting that there is a shift in background frequencies that occur on EEG as the disease progresses. So in conclusion, hopefully I've shown you that our mouse models of CLN3 disease do have network level excitability changes in the central nervous system, that both disease neurons and astrocytes contribute to circuit level disease, but it really seems that neurons are driving many of the phenotypes, much of the dysfunction that we are seeing. We saw that early correction via AAV gene therapy at P0 can improve function even without correcting storage accumulation, again, suggesting whatever CLN3 is doing to change synaptic function, to change network level excitability, might be separate from whatever it is doing that is resulting in storage accumulation. I've shown you that network and biochemical abnormalities arise really early in this disease, essentially at birth in CLN3 disease mouse models, which really is going to make us sort of rethink the paradigm what we consider disease onset in this condition. And we really need to start looking at our patients earlier on to really understand if we're missing any subtle phenotypes. This also really is important when we think about timing of therapies once therapies hopefully become available. Finally, we saw that circuit maturation is abnormal. So it's not just that the circuit is abnormal and stays abnormal. As the circuit matures, the normal maturation process doesn't occur, right? The hippocampus has to mature to be fully functional. And we're seeing that the CLN3 hippocampus just does not mature the way it should, suggesting that this really is a neurodevelopmental disorder. And with that, I'd like to thank all the folks in my lab who have done all this work. And I'd like to thank the funding sources, obviously, including the NCL-Stiftung, who have supported our work from very early on, and to recognize key collaborators, including Eric Marsh, Beverly Davidson, and Louise Tecador. Thanks.